I hope you already signed the safety sheet. Today is the first session after the orientation, which I'm going to talk about microbes in the environment and also broth cultures. For microbes in the environment, before we go to the experiment, you have to know that what is the inoculum and what is the inoculation. So the inoculum is the microorganism which we use to initiate a culture, and the inoculation is when we transfer the microorganism, microbes, to a new environment where they can grow. So we call this term inoculation. So for example, from the petri dish to a culture, from culture to a petri dish, from somewhere to somewhere else, which provides a good environment, good condition for microbes to grow. So what is a nutrients agar plate? So this is the solid growth media, which typically is sterilized, and it has the nutrients. The nutrients is important for bacteria to grow. This is the agar plate. This part is the agar side, and this is the lid. So always bacteria can grow in agar side because it has the nutrients, it has the agar, it has the water, and also we need to streak out on an agar side. I will show you how to streak on an agar plate. So for today, we are going to test 4 different areas in order to show that microbes are everywhere. So the microbes in the lab, in the human body, even in our face, in a natural environment, for example, the soil, and in the trees, in the leaves, and also even in an open air. So when you open the agar plate, put it on your bench for 30 minutes in a temperature or whatever you want. After you put it in incubator, you can see that the microbes grows. So it means that the microbes are everywhere. So typically today we are going to streak bacteria, the microbes, from these 4 different areas, and then test it to see that which part of these 4 different the microbes are more. So the first thing that you should do that is you need to label your agar plates. You have to label it on the agar side, because when we put it in incubator, I will show you what is the incubator and how you should put your samples in it. But when you put it in incubator, the agar side should be up like this, because we wanted to avoid the water condensation. So you have to label your plate. Always you are going to do the experiments like your name, your initial, your section, for example 801, and the location where we get our samples. So the first area is the lab. So we are going to get our samples from the lab. Okay, so what you should do is you need to touch a swab on an object and gently move the swab back and forth across it. So here is the sterilized swab. You need to open it and make sure you should not touch this part. For the lab, I will grab the samples from the bench. So we know that the bench is not clean, right? So we will rotate it completely. Then you need to streak out on a plate, back and forth gently, all over the plate, like this. After you are done, you should close the lid right away, and then put this swab back. Okay? And here is the biohazard materials. No glass, no sharps, just biohazard materials. You need to put it here, throw it away, and then move to the next samples. The next sample is human body. So I will grab samples from my hands. But first I'm going to wash my hands. So I will wash my hands for 30 seconds. Okay. So now my hands are clean, supposedly. Then I will grab another plate, label it again, AG 801 section, this time from clean hand. Grab another one. Hopefully there will be no microbes in my hand after washing. Back and forth until the end. Close the lid. Okay. So we need to do the same thing in a natural environment. I will go downstairs, grab some soil from the samples, from the environment, and then exactly do the same thing. Streak the swab on the surface of the nutrients agar plate. We need to rotate the swab over the media to ensure microbes are transferred. And the last thing is open air samples. So as we talked earlier, microbes are everywhere. So we wanted to show that even in the open air that we couldn't see with our naked eye, microbes are in the open air. What am I going to do? Again, labeling. Open air, AG 801. Open the lid and keep it for 30 minutes in the bench. After doing that, we will move all 4 samples to the incubator. So this is the 30 degree incubator. When you put your samples in incubator, you provide a condition for microbes to grow. So after 30 minutes are done, we have 4 samples from 4 different locations. So you can move it and put it in incubator. Here is the incubator. Agar side up, like this, and then you need to close it. And we will see the results in 2 days.